So on Wednesday, Allison, I believe you asked me about can other viruses incorporate their genomes into the genome besides just type 6 and type 7? And so I did some more research, and yes, some viruses that have, so some type 1 and type 2 viruses that have DNA have mechanisms by which they can get their nucleic acids into the nucleus without the reverse transcriptase, that just have mechanisms to get that material in there. All right? Okay, well, I think have either 3 or 4 framing questions to deal with, and then we'll be done. All right? All right, so the next one, how do plant viruses affect humans? All right, so this first part, we'll answer it this way, and then I'll give you a concise answer to this. So viruses tend to be very host specific. Most of them, in fact, tend to be species specific. And so the dominant idea of viruses is basically this. You have bacteriophages that infect bacteria, you have plant viruses that infect plants, and you have animal viruses that infect animals. And until 2 years ago, there were no examples anywhere of any alteration to this. So I actually gave you a really wonderful tool to organize viruses further, right? So you could do it by structure, you could do it by the type of nucleic acid, and then you could do it by the type of host. Is it a phage? Is it a plant virus? Is it an animal virus? Starting about 2 years ago, we started to realize that there are phages that infect primarily bacteria that actually have animal DNA in their genome. Like, that is really weird. For instance, there's a bacteriophage called WO, is the phage name. This is just bonus information, but it's a cool story. It's called WO, W-O, really wonderful name for the virus, and it infects a type of bacteria that infects all kinds of different insects and spiders. And they found the gene for black widow venom in the virus's genome. The only way that it could get there is if, in addition to infecting the bacteria, it also in some way infects the spider cell as well. Not only did they find the gene for black widow venom in the virus, but they actually found that the virus gave that gene to some insects, and now that the insects are making black widow venom. Right? Isn't that awesome? And so you have some species of stink bugs that are making black widow venom, and they were given that gene by a virus that supposedly only infected bacteria, but actually infected black widow spiders at some point, and the stink bugs that now have the capability of making black widow venom. And you're like, wow, this is kind of awesome. So viruses don't seem to be as, picky as we think. Although it's still fairly rare to find examples of viruses that this isn't true for. This is still the norm, but there are some exceptions. So this is the norm, no exceptions. Now this is the norm, few exceptions. Okay? So how do plant viruses affect humans? Indirectly. Okay? Indirectly by impacting our food source or our food source's food source. Right? Let's say you raise cattle and there's a virus that infects the wild grass that your cattle graze on. So then that impacts your cattle, and then it impacts everybody that eats your cattle. And it's sad, right? Because then the cost of beef goes up, and then you complain even more when all you want is ground beef and you have to pay $5 a pound for it. And you're like, really? Is this a ribeye? No, it's just ground beef, and it's just sad, right? So indirectly, although here's a cool story. This is also bonus, but it's a good little piece. It's a good little nugget of truth. If you determine all of the viruses in an organism, that's called the virome. The virome, fancy term that just says all of the viruses found in an organism. The human virome is like 95% plant viruses. So 95% of the viruses you find in humans are plant viruses. Like, that's really weird. What are they doing there? And they they came from your food. And then they just stick around. Like, what are they doing? Who knows? But they're there, and they're living, and I'm guessing that they're enjoying themselves. I was going to say enjoying life. They're not living. I lied. Oh my gosh. That was terrible. They are not living. They are existing. They are existing. They are not living. Wow. Yeah.
From the quiz, actually.
Okay.
So the fourth question, I think.
Yes.
Where it told us which one we not categorize them by.
Yes. I don't remember that question. I mean, I remember the question here. Yeah, give me the choices. Give me the choices.
It was
By structure. Yeah, by nucleic acids.
By disease.
Oh yeah, we don't organize them by disease because we didn't talk about that, and it would be a very bad way to organize viruses. Because then you're either like this disease only goes with a single virus, right? It's not going to do you any good of organizing something that way. It would be like, let's say all you have, every shirt you have is both a different color and a different pattern. And you're like, I'm going to organize this by color. And it's like, okay, you're just taking every individual shirt and putting it into its own category. You're like, better yet, I'm going to organize it by pattern. But then you still have the same issue, right? And so that's what would happen if you tried to organize viruses by disease, because a virus causes a specific disease. And so it wouldn't actually do you any good to organize that way. Yeah?
What is virome again?
Virome is the collection of all of the viruses in an organism. Yeah, yeah. This is a good question. All right. So then just kind of some general characteristics of viruses that we need to talk about here. So you have a characteristic that's common in both plant viruses and in bacterial viruses, what we call bacteriophages, and that's for them to hide out in the host genome. It does happen in animal viruses as well, but we know a lot more about it from studying bacterial and plant viruses. And so I just want to mention that a cycle where the virus is inserting its material into host cell, making new viruses, leaving host cell, that's called the Lytic Cycle. And it comes from the word lysis, which means to break open. And so the Lytic Cycle, the viruses are actively replicating, infecting cells, making new viruses, erupting. Okay? But the Lysogenic cycle, that's where they go dormant and they hide out in the cell's genome. And in bacterial viruses, we call this a prophage, where it's just hiding out in the bacterial genome. This happens a lot in bacterial viruses, bacteriophages. It happens a lot in plant viruses as well. That's why I put it there. But I just wanted to reference, we haven't actually talked about these points before, and this was an okay question to actually deal with it. Yeah, animal and plant viruses can have this lysogenic phase as well. Now, the reason why plant viruses don't directly impact animals is because in order for a virus to get into an animal cell, it has to be able to bind to targets that are only on animal cells. And so if the virus binds to plant cells, it has machinery to bind to targets that are on plant cells. In order to actually infect an animal cell, it would also have to be able to bind to targets on animal cells. And now you're adding a lot more capability inside of a virus in a very small package where there's very little room for instructions to build machines. Okay? And this is why it's so fascinating to find that there are bacteriophages, which are the smallest of all of our viruses, that actually have DNA from animals inside of their genome, because there is not very much space. And it makes you wonder where they came from in the first place. Makes you wonder. All right, and then so these are the processes by which the viruses can enter into a cell, whether it's an animal cell, a plant cell, a bacterial cell, although animal cells and plant cells have more sophisticated processes. So this process we'll talk about later in this class, receptor-mediated endocytosis. Basically, it's just a fancy way of saying that the animal cell or the plant cell brings the virus in. So once the virus binds, the cell does the work of bringing the virus in. And that's ideal for the virus, because that requires less machinery. right? All they need is a machine to bind to the cell, and then the cell does the work of actually bringing them in. If that's not how it works, the viruses either have to inject their genetic material into the cell, or they need to fuse with the lining of the cell. Okay, any of those, these are very sophisticated processes, even just binding to the correct cell. And that's what oftentimes prohibits plant viruses from infecting animals. And that's why they impact us indirectly, but can cause huge economic losses, like the tobacco mosaic virus. Huge economic losses, yeah.
Why would the cell bring a virus into?
It's a wonderful question, because it doesn't know that it's a virus. It just receives a signal that something is attached to that receptor. And it's a receptor that is necessary for the cells to bring in normal material. And so it's another example of viruses hijacking normal machinery to end up getting the host cell to do what they want. So a lot of times the viruses will target receptors for like specific proteins that cause the cell to go through replication. So not only does the virus bind something that tells the cell what to do, but it tells the cell to start making copies of itself. So all of the machinery is in place for then the virus to hijack to make copies of the virus instead of allowing the cell to make copies of itself. And it's so wonderful. Binding a receptor that's already there, and then now the cell actually starts another process that the virus is going to take advantage of. It's wonderful. It almost seems like viruses were designed to have this impact on the host. And then so there's a question of, man, how does that happen? How does that happen? We'll talk more about that during our lecture break. Yeah.
So, once the virus enters into a cell, is it game over for the cell?
Not necessarily. It depends on a number of factors. So sometimes the virus will enter the cell and it just doesn't work properly to make copies of itself. Either the viral DNA or viral RNA is damaged in some way, so it can't make copies. Or, a lot of cells, they recognize foreign material even inside the cell, and they'll just shut the whole process down. Cells will oftentimes kill themselves after a virus infects the cell because they know that they're infected, and they'll kill themselves before they make copies of the virus.
Okay.
So that's not necessarily, plus the virus may just go and enter into the host cell's DNA and sit there for generations.
Okay.
Yeah.
So with endovirus,
Yes.
is it the same sort of action as endocytosis, or is it more of like its
Yeah, I mean, endocytosis comes in like two main categories. You have receptor mediated, where you get a big pinching off, or you get something like pinocytosis, where you just get a fusion. But yes, yes.
Does it stay fused to the membrane though, or does it enter?
The membrane of the envelope stays fused to the membrane. The rest of the virus enters in when it's absorbed, and that's something that's similar when cells absorb like pockets of material in a vesicle or something. Yeah. Yeah.
So what's the difference between like having a latent virus in you and having an asymptomatic disease?
Usually the reason it's asymptomatic is because the virus is latent. But it could also be that whatever it's doing is not having, so the virus may be actively replicating, but it's not leading to any pathology, right? So the virus is actively lytic. It's making copies of itself, but whatever cells it's blowing up on its way out, it's not having any impact on pathology. But that's rare. Most of the time, the reason why it's asymptomatic is because the virus is latent. It's hiding out somewhere. And so that happens a lot with like the chickenpox virus, where your immune system will get control over it, but you'll have that viral DNA sticking around in your cells, and maybe 35, 40 years later, something triggers it to come out again, and it erupts, and it's changed enough in the mistakes of your own cells copying the virus DNA that your immune system doesn't recognize it anymore. Yeah. Yeah. It's awesome. And then you get shingles, which I hear is incredibly painful. I don't know. I watched my dad deal with it, and he's not a complainer. So I was like, wow, this doesn't seem painful at all. But I don't know the way he felt, just the way he acted. All right? Any other questions, thoughts, concerns? We're going to do a lecture break, and I know we've only done one question, but we did a quiz and then did one question. And so what I want you to do is this. I want you to, with those around you, come up with all of the features of a virus that implies that they were designed to do what they actually do. Okay? So all of the features of viruses that imply that they were designed to do exactly what they do, and that's to hijack a host cell for its own replication. Okay? So come up with all of the features. Another way to ask this question is, what is actually necessary for the virus to replicate, right? Because it's got to hijack the host cell in order to replicate because it's not living. What are all the things the virus has to do? All right? Just make a list, have it all in one place, talk about it with those around you, and we'll talk about it collectively.
Any initial thoughts about that's what my how they're shaping
All right. So, what are some of these things that a virus has to do, or these elements that seem that they are perfectly designed to do what it is that they do? And that is hijack a host cell for their own replication. Yeah, Jack.
So like we were talking about just their structure and their design is meant to like attach to things.
Yeah. Especially the head and tail viruses. I mean, they have a whole portion of their structure that's just like a claw. Yeah, just like, wow, that looks pretty special, right? It's like, where did that come from? That seems so wonderful. Yeah.
Yeah, we're talking about the same thing, how different viruses have different viral receptors.
Yes.
Cling on to specific types of cells.
Yeah, right. Even not just the head and tail viruses, but all viruses, they have to recognize. So it is a non-living entity that recognizes a specific receptor and binds to that receptor. And it's so wonderful. Because you're like, is it really recognizing it, or does it just happen to be in the right place at the right time? Well, the only way that that would be true is if there were literally hundreds of billions of virions, right, those viral particles, and they just happen to be everywhere. But we don't find that, right? They happen to only be in the place that you would expect them. And you're like, well, what about those plant viruses in the human virome? Yeah, but it's from the plants that we eat. So like I eat different plants than somebody from another part of the country and somebody from another part of the world, and they have plant viruses from the plants that they eat. So it's like they're exactly where we'd expect them to be. Like, why are they hanging around? I don't know. They're not like eliciting the immune system to react. I mean, they're just kind of hanging out. They just get stored up. Maybe it's because our bodies are designed to actually facilitate the survival of viruses, because viruses have to actually enter into our cells to get our cells to function properly. Maybe. Just some food for thought. Allison.
We're also talking about the fact that they have receptors, but then also the DNA that can be incorporated
Yeah.
into the cell.
Yeah, they've got to have they've got to have the code, the blueprints for our cells to build viral proteins, right? Where I put that little process up on Wednesday, I think it was. Yeah, it was on Wednesday, because on Monday we didn't talk about viruses at all. And you had I just wrote translation, but you have to go from RNA to, viral protein. but it's got to have the blueprint to do that, right? Our cells don't know what to build. The virus has to show them what to build. And it's so wonderful. It's not a living organism, and yet it can hijack a very sophisticated cell and tell it exactly what to build and when and how much. Yeah.
So if we're like saying that it has like cell receptors and you can like target specific cells and all this, does it then fulfill the requirement that it can react to like stimuli, or is it like different than that?
Yeah, it's an interesting question. I don't know. I don't know the answer to that question. Does its recognition of particles qualify as responding to stimuli? I don't know. Yeah, I don't know the answer to that question. I think you could make a pretty good argument that it does. Although, so maybe it does have the respond to stimuli characteristic of life, but it surely is missing other characteristics of life. Yeah.
I thought on Wednesday you said that it doesn't necessarily respond, but it can react to a trigger. Is that the same thing, or is it different?
Well, it doesn't move freely, right? So it doesn't, and then so you're like, well, how does it get then to the right place if it doesn't move freely? And I don't know. I mean, it's carried to the right place because of the way that it functions. It's like if they need a new host, you are perfectly willing to give your viruses to anybody that comes around you, right? Especially viruses that are airborne pathogens, but even those that aren't, right? You're like, you share a cup with somebody, or you hold somebody's hand, right? And it's like, man, next thing you know, you're sharing your influenza virus with everybody around, and everybody just feels wonderful, right? Everybody just feels wonderful. All right? Or, oh gosh. What was the, oh, never mind. Never mind. I'm sorry, my brain's not working well today. I didn't sleep last night, so.
They can disguise themselves too, like sometimes when they emerge from the cell,
Yeah.
some membrane with them.
Yeah, they'll carry some of that cell. I mean, that's just wonderful, right? It's like, when did it start? Because they had membranes. So let's say somebody is so kind as to give you their virus, right? So their virus enters your body with their cell membranes on it. So your body recognizes it as foreign, but before it actually has a chance to destroy it, the virus does what it's designed to do and get inside your cell, and shed. That's what happened. Cell and shed the membrane from the person you got the virus from. But then by the time it leaves your cell, it has your cell membrane on it and is protected from your immune system. But then when you give it to somebody else, it's got your membrane on it. But again, before their immune system has a chance to attack it, it does what it's designed to do and gets in one of their cells and sheds your membrane. And it's just so wonderful. It's like, yeah, it helps it to hide out from your immune system, but actually complicates its ability to hide out from somebody else's immune system. But because of other aspects of the virus, they get inside of a cell before your immune system reacts to their presence. That's just so awesome. It's like, where did these things come from? And why do they function this way? They're highly sophisticated machines that do what they do extremely well. Extremely well. This is just a wonderful thing. This is just a wonderful thing. We're gonna put that thought away and for another day. Did you have something, Mia?
I was just gonna talk about how like the DNA can like become incorporated.
Yeah. And then not only can they just evade your immune system, but they can actually become part of your cell for generations, right? And then something happens later on and poof, they come out, it triggers their growth. So these are the lytic and the lysogenic stages. Here's lytic. Virus puts its genetic material into host cell. Host cell makes new virus particles. Those virus particles assemble into new viruses, and new viruses blow up, host cell and go and find new cells to infect. Here's lysogenic. Virus inserts its genetic material into host cell. Genetic material gets incorporated into host genome, and it just stays that way until something triggers it to leave. And it's like, what triggers it to leave and why? And it's different for every virus. And sometimes the particular trigger that causes it to come out and to start replicating can happen, but it only works when it's in the presence of something else. Like the trigger is there, but the host cell also has to be maybe malnourished. Yeah.
Which viruses are more deadly, like the ones that wait and then come out, or the ones that come out immediately?
Yeah, that's a good question. I mean, viruses cause most of their issues when they're lytic, and so particularly virulent viruses, which are those that spread rapidly and tend to cause pretty extreme pathology, is they do it while they're lytic. The viruses that are just replicate like crazy, because most of what you experience pathology-wise is your immune system trying to fight something that's out of control. Now, there are some exceptions to that if the virus is infecting, say, like T cells, which is what HIV does, and then your immune system loses its ability to fight other infections. But most of the time, what you experience as far as like, the body aches, the sweating, the incredible fever, right? All of that is your immune system trying desperately to fight something that's actively growing inside of you, but only growing by using your material to do it. Yeah.
So, the virus that can be triggered
Yes.
by something, will it always maybe be triggered by the presence of another illness or maybe like stress?
Yes. It varies. It varies. And then if it sticks around long enough that as your cells are replicating, its own DNA gets damaged, it can get to a point where it can never come out. And this is like it's just so weird. The part of our immune system that actually fights viral infections looks like it originated as a virus that inserted into our genome and then lost the ability to leave because it mutated while it was in the genome. And then now our immune system uses it to actually fight viral infection. And then it's like, well, that's really weird. Why does it work that way? Why weren't we always designed to fight viral infection? Maybe it's because they were designed to actually do something good, and then when they became pathogenic, we needed a way to actually fight viruses. And with a virus itself gave us the tools necessary to fight other viruses. I mean, this is just awesome. Yeah.
So are there portions of our DNA
that end up in the viruses?
Or our DNA that, like, because they're viral DNA that's not being used, like, we just don't, it's just like extra DNA that we don't actually use it?
Yeah, you find very little of that. So that was a very popular idea all the way up until, I don't know, about 10 years ago, that we just had huge amounts of our DNA that was just junk. That came from various sources. Maybe a lot of it came from viruses that got stuck, and then it's not functional. It doesn't give any kind of a code. But starting about 10 years ago, and especially 6 years ago now, in 2012, people have been publishing work that suggests that the absolute biggest piece of DNA that you find in our genome that doesn't do anything is like 7,000 bases. Which you're like, wow, that's kind of a lot. But when you've got a 3 billion, base pair long genome, I mean, that's one. Let's see, that is 1/1,000,000th of. How would this work? So 1% of 3 billion would be 30 million, and so 7,000. I don't even know. I mean, that's like this is like a very tiny portion of our genome is the whole thing. I was gonna do the math, but my brain's not working today. And so, I mean, it's just shocking, like how much of our DNA is actually active, including parts that look like they came from viruses, that look like they were inserted into our genome from foreign material, and yet they're active, and our body uses them to carry out its everyday function. It's just weird. Yeah.
Question about the Lytic cycle.
Yes.
So for the nucleic acid.
Yes, with the viral or the host. So this is showing you it in a bacterium, just because it's simple. Their DNA isn't packaged in a nucleus, so the image is simpler.
Yeah, but it stays out. It doesn't combine with the host DNA. So how does it make more copies of itself if it stays apart?
Oh, because the machinery to replicate this is inside of the cell. It's the same machinery that would replicate the bacterial DNA, but it's in the cell, and that machinery is available for replicating that. So at the same time that it's replicating bacterial DNA, it'll also be replicating viral. But the viral genome is so small that it replicates so fast that it very quickly overwhelms what's happening here.
Okay.
So let's just say for kicks and giggles that this is E. coli, which happens to be the bacterium we know more about than any other bacteria. If this is E. coli, it takes about 20 minutes to replicate the E. coli genome. It would take probably 35 seconds to replicate this viral genome. Maybe less, maybe 3 seconds. Okay, and so then the amount of time it takes you to do this, you're going to replicate hundreds of thousands, potentially, of viral genomes that then are all going to get packaged. Because then at the same time that this is being replicated, there's also machines that are taking the code and building proteins with the code and are building new viruses.
Gotcha.
Yeah.
A certain illness actually
Yes.
often, 'cause
Yeah.
like 4 or 5 years ago maybe, I had pneumonia really bad.
Uh-huh.
And like I have not gotten sick since.
Yeah. It's interesting. Do you know what the pathogen was that caused your pneumonia, whether it was viral or bacterial or fungal?
Hospital, so they didn't.
Yeah, that's disappointing because it'd be interesting to know. Yeah, pneumonia can be caused by almost any pathogen. Like, so you've got viral pneumonia, bacterial pneumonia, fungal pneumonia, parasitic pneumonia. But yeah, it'd be interesting to. But yeah, I mean, there are illnesses that do prevent you from getting other illnesses. Absolutely. So, and then there are diseases which are a little bit different, like not caused by pathogens, that prevent you from getting other diseases as well. Yeah, so it's very difficult to have both, like to have HIV and HPV. They've shown that there's some very strange relationship there, that people with HPV seem to be less susceptible to getting HIV than people without HPV. And then there's Sickle-Cell Disease, which is not caused by a pathogen, but it makes it very difficult for you to get malaria. Not impossible, but very difficult. Yeah.
Well, my friend that has HIV that takes the medication for it, they don't ever get sick, and they like create a better thing in their house that like less people get sick. And I found that pretty common because I have like a lot of friends from where I'm from in South Africa that had it, and a lot of the homes were kids that had HIV and were on this medication. What medication is that?
It's probably AZT, which is attacking reverse transcriptase.
So it
And
affects-
well, so it's going to attack to any retrovirus is going to be attacked in the same way and is going to be treated by that. So any virus that requires its genome to be inserted into the host genome, the AZT is going to combat that. But you have a number of factors. So you have what's called the hygiene hypothesis. And it's coupled with another hypothesis called the old friends hypothesis. And both of them basically try to describe that in order for your immune system to behave correctly, there are certain illnesses you have to get. Like there are certain sicknesses that your immune system needs in order to mature properly. And so what you find is that people in areas where they typically do get certain illnesses as they're developing, their immune systems behave properly as long as malnutrition isn't an issue. But it just happens to be in a lot of those places, malnutrition is also a big issue, which if you don't have enough energy, like your immune system's not going to behave properly. But it's interesting. So what you find is like people in tropical areas, period, they tend to get certain illnesses and their immune system behaves properly and matures properly. And so that if they stay healthy, like they're not malnourished and they don't, get some kind of novel, weird, strange disease, they tend to get sick way less than people that grew up in developed countries. And they tend to have way fewer cases of allergies and autoimmune diseases. And it seems to be like your immune system needs certain things in order to mature properly. Yeah.
So if then, I was going back to pneumonia.
Okay.
If it was like bacterial versus fungal versus
Yes.
viral, would that, would any certain case of pneumonia cause you to be more susceptible to sickness or less susceptible to sickness?
Well, they're all gonna do different things as far as your immune system is concerned, because the way your immune system fights a bacterial infection is different than the way it fights a viral infection. It's different than the way it fights a fungal infection. Way different than the way it fights a parasitic infection. And so yeah, they're all going to have different impacts on your immune system. Is there any one that is going to be more or better or worse at actually preventing future illness? I don't know. I don't know the answer to that question. These are things that you have to kind of study with what you call correlational studies, because there are ethical issues purposely infecting people with illnesses to see how that's going to shape their immune system. People don't really like that, and it's hard to get research like that approved. And so you have to do it usually with like correlational studies, like, hey, these people that had this illness as children are 7 times less likely to develop this, you know? So it's like, is that really what's causing it? I don't know. Or does it just happen to be? So let me give you an example of this, and we'll actually finish here. Yeah, we'll finish with these pictures and with this. So there is a extremely strong correlation between the number of libraries in a city and the amount of crime in a city. Like, there's a super strong linear correlation. As the number of libraries increase, so does the rate of crime. But I don't think anybody would suggest that the number of libraries is increasing people's likelihood to commit crimes. It just so happens that there's other things involved, that where you have a lot of libraries tend to be in places where there's a lot of funding for trying to combat certain things that happen in inner cities. And so you have that. And so it's like you have this issue whenever you're doing correlational research. It's like, yeah, there is a very strong correlation. Oh, we still have some images to go through. We're this story and some images. I'm just not going to do any more framing questions. Relax. We got some time left of class. It's like, yeah, I mean, you can say that somebody that had this type of pneumonia is significantly less likely to get this disease, but you can't say that it's because they had that particular illness. Unless, of course, you did a research project where you deliberately gave people that illness, controlled everything else, and then demonstrated that they were incapable to be infected with something else. But there are some ethical concerns there. Even with just doing that with mice. You're like, oh, we'll just do it with mice. There's some ethical concerns involved. And so here's an example of the Morsella Zoster virus. This is a type 2 virus. Got single-stranded DNA, right? Double, single, 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 double. It's got single-stranded DNA. And, so never mind. It says right here double-stranded, which would make it a type 1. Disregard that I called it a type 2. So this is a type 1 virus, and this is one where it can get incorporated into the host genome with mechanisms that it has. Here's HPV, and HPV is one. This is where I mentioned that people with HPV seem to be less susceptible to contracting HIV than people without it. And so there's some very interesting thing going on in terms of how this virus shapes your immune response. And then HPV is a virus that can then cause cervical cancer and other type of reproductive system cancers, but it tends to be asymptomatic. And yet there's something going on with how it shapes how your immune system matures. Yeah.
The previous image, you mentioned the type. Is there a way to figure out what type of DNA?
Yeah, by the type of DNA, because that's what the Baltimore system is entirely built on. So if it's double-stranded DNA, your only options are type 1 or type 7. And if it's type 7, it's a retrovirus, right? Because only type 1 and type 7 have double-stranded DNA. But then type 4, 5, and 6 all have single-stranded RNA, right? Yeah.
What other types of virus, like, which type is the hardest to identify?
You mean as far as characterizing?
Yeah, like identifying that this is the virus that's making you sick, or this is causing you to do this.
I mean, it really just it depends on more of the viral life cycle than of the actual genetic material that it's made up of. So if the virus has a tendency to go latent and to hide out, those are viruses that are just very difficult to find and diagnose. So, yeah, it's more a question of the life cycle of the virus versus the type of nucleic acid. All right, it's 12:37. We don't have time to go through the next framing question, so now we'll be done. Have a wonderful day. If you want the last 3 minutes of class, hang out, we can chat. If you don't, ride off into your 3-day weekend. Yes, sir.
I got a question for you. What was the, I didn't hear the response.